Hello guys, today we are going to see our next module which is the marine biotechnology, and within this lecture we will see how exactly oceans or the marine environment or biodiversity and how it is improving biotechnological tools and how these different resources are helpful in for the mankind or for human life. So we are going to see what do we call this lecture in detail. So before we see, we need to understand what is oceans and what it is. Basically, life started in the oceans or in the sea. The Earth's surface is mostly oceans. You all know that 70% of our surface of Earth is covered with the water, which has the most ancient and most diverse forms of life. Thus, we can say that the marine environment is the treasure trove of biological and chemical diversity among all types of ecosystems. So, it has a wide variety of living organisms. We can take example like from bacteria to eukaryotes, as well as some unique chemical compounds, some other very precious chemicals, minerals that we can get out of the oceans. Now, if you see in other aspects, there is a great importance to oceans because of we can get ample amounts of medicine, nutrition, cosmetics, agriculture, and other industrial products. Biotechnological applications can be used to harness the potential of marine environment for human benefit and their fundamental biological processes. See, guys, marine biotechnology, it is the creation. What if we elaborate the term marine biotechnology, we can say that it is the creation of products and processes from marine organisms through the application of biotechnology, molecular and cell biology, and bioinformatics. It is the field of science that deals with oceans exploration for development of new pharmaceutical drug, chemical product, enzymes, and other products and processes. It also deals with the advancement of aquaculture and seafood safety, bioremediation, biofuels, and many more, like any other. If you see the applications of marine biotechnology in terms of medicine, then over 2000 years, extracts from marine organisms we were using as the medicine. The recent footprints of these knowledge is in 19th and 20th century. The cod liver oil is one of the famous nutritional supplements in human. If you take an example, if we say, if we go back in 14th and 15th century, the Portuguese or the European different expeditions, cruise expeditions or marine expeditions, they just do business to hunt liver oil or to hunt these whale species to get the cod liver oil, and they just sell it in the markets, local markets, or even in the international market, and to get many much amount of money out of it. Now, why it is the important? The oil, the liver oil is very much important. It is the source of fatty acids. It's a source of vitamins, the beta, omega-3 fatty acid, and everything. Like that's why it is also acting as a staple food for some in the, or it is the one of the ingredient in food in many more countries. So it was only in the middle of 20th century when scientists started to systematically navigate oceans for the medicines, like which oceans are actually very well diverse and where we can get the large quantities of such organisms. When scientists were studying the defense mechanism of sea creatures, they discovered the vast defensive chemical weapons of the organisms. It's a very natural tendency when single organism get attacked with their predators, definitely they defend them or otherwise they attack also on their predators to save their own life. In 1950s, Ross Negrelli from New York Zoological Society extracted a toxin called holothurin. Now, what is that holothurin? They extracted it from Bahamian sea cucumber species. The scientific name of that species is Actinopyga agassizi, which showed, okay, this is the name of that species, which showed anti-tumor activity. That means it shows the anti-cancerous activity in mice. When they worked on a mice, they observed this anti-cancerous activity of this toxin. Okay, the holothurin, it is the medicine or it is the toxin, was not commercialized, but the number of potential bioactive compounds from the oceans spiked up, and more are being discovered every year. Scientists have used this biotechnology to make copies of marine compounds. Now, if we come to know the actual compound, how exactly these compounds, what is the structure of like holothurin, we can mimic that compound, or we can take the activity, how exactly this compound acts on these abnormal growing cells, where they attaches and where exactly they kill these abnormal growing cells within the organism. We can mimic such a compounds, and we can what do we call, we can make it into the clinical research, and then we can ultimately make medicine which is safely inserted into the human body. So the biotechnology field is helpful over there. Okay, so back to our topic. Some have been commercialized, some of them are already commercialized, while others are undergoing clinical and preclinical test. So there is a table, we'll see, that summarizes some of the commercialized medicines derived from marine organism. Other marine-derived drugs are still in clinical trials, and these include cytotoxic compounds, okay, like bryostatin 1 and the dolostatin derivatives of soblidotin and synthedotin. Now, aside from these drugs, more products are in preclinical pipelines. The number of marine compounds reported is increasing every year, with over 1,000 new compounds with the varied potencies for, definitely, for some new diseases, like some disorders are there which we cannot able to cover or we cannot cure with the help of any medicine, but their prognosis we can make a large period of time to get their prognostic effect or their illness. We can add these compounds into the body or into the organism to overcome this disorder. The biological functions added to the pipeline every year. This is the list of these drugs which are currently in the market, and they have a very good market values, and we are also purchasing them for different purposes, like cephalosporin is one of the antibiotic drug which is isolated from the cephalosporium fungi. Okay, and this antibiotic, we generally widely we are using this antibiotic in day-to-day life, in agricultural or clinical research, everywhere. Then squalene is also one of the antibiotic which we have already discovered from spiny dogfish. The scientific name was Colus acanthus. So this antibiotic is also commercialized nowadays. Okay. Next one is vidrabine, which is isolated from Caribbean spongy. Okay. Tethia crypta is one of the species of the spongy, where vidrabine is the antiviral drug that we have isolated. Then cytarabine, then zinconotide, okay, then trabectedin or eribulin mesylate. This is one of the two drugs which are really helpful and showed a very great efficacy in anti-cancerous activity. Different types of drugs are there. There are thousands of drugs, but these some of them are really very interesting that we need to understand. Apart from the medicines, apart from these drugs, we are also extracting secondary metabolites from the marine sources or from the oceans. Now, what is meant by secondary metabolites? There are around, from one of the publication from where I have taken this data, Within that publication, 71 secondary metabolites they have isolated from marine fungi, actinobacteria, and they reviewed this study and summarized that around 45 to 27 secondary metabolites they have isolated. The fungi basically are from genus Aspergillus, Penicillium, Neosartoria, right? Then Dichotomomyces, then Paradendrophylla. So these are some fungi. Then Wester Dicula. These are some genuses of fungi where they have isolated so many secondary metabolites that could have a activity of antibiotics or some anti what do we call the healing activity, or might be they are having some analgesic property like this one. Okay, so these are the secondary metabolites which we have isolated. Out of that, actinobacteria are represented by genera like Streptomyces or Nocardiopsis species particularly. The secondary metabolites from marine microorganisms show potential cytotoxic agents. Okay, they are having a very good property of like anti-cancerous property. So particularly those sourced from the fungi Aspergillus and Penicillium. Okay, uh, these actinobacteria shows a very good secondary metabolite production. We can culture these microorganisms when we get them into the laboratory. From primary culture to mass culture, we can cultivate them and we can isolate these secondary metabolites in large amount. And that is the actual use of biotechnology. Okay, most of the compounds are found to be indole alkaloids or diketopiperazines. So these drugs, these compounds shows a very good activity against any bacteria or pathogens or even the parasites also. These peptides or alkaloids from 71 secondary metabolites are the group with the most potential for novelty related cytotoxic activity. Specifically, these are diketopiperazines. These are cyclic compounds for peptides and indoles for alkaloids. Their high potency can be explicated as the effect of introduction of halogen-based functional groups in indole alkaloids and presence of sulfide groups. Okay, in these diketopiperazines. It is expected that this provides more insight into accelerating drug discovery, research, and development. If you see these 71 compounds, some of them I have listed in one these list. There are the different secondary metabolites. Out of them, certain amines and amides are there, indole alkaloids are there, acetogenins are there, cyclic peptides are there, microlids are there, sequestered pins there, then phenazines are there. Like these compounds, these I know that as a TYBSc, you peoples might be don't know some of the some names out of these compounds, but these compounds they are very effective and very important in activity in certain metabolic reactions. Some chemicals inhibit some reactions, some chemical boost up several metabolic processes, and these processes these are really important in human metabolism. So just to find out where the key role of these compounds are there and to prepare medicine and check their activity or check their efficiency. These compounds has been isolated from several microorganisms as well as invertebrate species. And how much compounds or how much percentage of the compound we are able to extract them out of these organisms, this percentage is located here. The metabolite from the microorganism is the most cases quite different from those produced by invertebrate host. Nitrogenated metabolites predominates over acetate derivative. Then terpenes are uncommon. So among terpenes, basically we can found them in the plant derivatives or plant basically product. Okay, so these is the alkaloid compounds where we can get from these marine organisms, and they shows a very good activity indeed. Okay, then next if you see barophilic organisms. Now what is meant by barophilic organism? Barophilic organism means those organisms which can sustain or live in very heavy pressure. See guys, our oceans our oceans is very deep. The maximum depth of the our ocean is about 11 kilometer, near about 11 kilometer. I cannot tell the exact, but 11 kilometer, and that is present near the Japan region. So these basically in the Japan, nearby the Japan coastal area, the region is called it as Mariana Trench. Over there, the atmospheric pressure or the pressure of the ocean is so tremendous that any even submarine will squeeze into the bottle like a shape. The whole submarine will be like the bottle or can like a shape. It will be squeezed up to that volume because of heavy pressure. And at that pressure, there is a big question that whether any organism can live. Yes, of course, there are organisms, but these organisms have very high tendency to live only up there. If we grab them and if we get them outside the oceans, and if we want to culture them on a land or above the ocean, then definitely they will die because it's their habitat. So these organisms, or basically microorganisms, which is present over there, these organisms we called it as barophilic organisms. So barophile is an organism that needs a high pressure environment in order to grow. Barophiles are the type of extremophiles. Now, what is extremophiles? Extremophiles are these organisms or microorganisms which survives in very extreme condition. Now see, this heavy pressure is the extreme condition if we compare ourselves with them. But for them, it is a natural condition. But for our reference, we have named them as extremophiles. An example of high pressure habitat is the deep sea environment, such as ocean floors or D lakes, where the pressure can exceed up to 380 atmosphere. Another is the subsurface rocks with the high lithostatic pressure. These over there also heavy pressure is there. Barophiles that cannot survive outside their high pressure habitats are referred to as obligate barophiles. Those that can live at high pressures and is less extreme environment are referred to barotolerants. Okay, so these organisms we can call them as barotolerant. That means they can tolerate very high pressure. One of the organisms is named as Halomonas salaria. It's a gram-negative proteobacterium, and it is the example of obligate barophil. It needs a pressure of about 1000 atmosphere. Many barophiles are sensitive to ultraviolet rays. Why? Because they actually throughout their lifetime they are unable to get even visible light. How could they get ultraviolet light over there? Because just after a few meters of the surface of oceans, there is a complete dark patch of oceans comes up where no photosynthetic activity at all possesses. Okay, so whatever photosynthetic activity is there, that only happens where the sun rays or solar energy or solar radiations reach up to that level. So the many barophiles are sensitive to ultraviolet rays and are susceptible to UV radiation. Okay, they basically lack these what do we call the essential mechanism of DNA repair to counter the effect of UV radiation. So they are lacking these DNA repair mechanisms, which is we have all the organisms on terrestrial or terrain or on the surface of Earth. They contain this DNA repair mechanism, but these organisms are actually don't have this because they are actually never faced UV radiation. Thus, many of them grows in the darkness. Because of this, they also tends to be a psychrophilic. That means they live under cold temperature. Now there are three terms. Let me tell you. Within the extremophiles, these organisms comes up barophile, then thermophile, then psychrophile, and the other one is halophile. So halophiles they can tolerate heaviest salt condition. Then thermophiles are they can tolerate a very heavy heat condition, and ultimately saprophytes, where these saprophytes they can tolerate a very cold temperature. That means they can live up there about 2 to 3 degrees centigrade below 100 meters of the depth. Another example of barophils is the xenophyophores, okay, which is the single-celled eukaryote found in the deepest ocean trench, that is about 6.6 miles below the surface of these oceans. 6.6 miles below the surface. The term barophil basically is coined by Zobel and Johnson, okay, in 1949. Excuse me for my these some spelling and sentence mistakes are there. Barophilus bacteria, they grow preferentially or exclusively or moderately in a very high hydrostatic pressure. Adaptation to high pressure, they have this capability. Adaptation to high pressure, they have an increased binding capacity of these enzyme. The polyunsaturated fatty acid in the membranes are there, okay. The pressure controlled gene expression is there. If the pressure increases, they have a very good gene expression during that time. If they release such a proteins that can save their genetic content apart from this pressure. Now next part that we need to see, seaweeds and the removal of metal pollutant. The environmental metal pollution is the serious public problem currently, because we are getting so much metal items in our home, and once we get once we use them up to their dead end or their life, we throw it them out of our home or in the garbage, but we don't take serious issue how much garbage we are producing, how much what do we call solid waste we are producing. It's the very big problem current nowadays. It has become an issue leading to the research in effluent remediation area, the wastewater system or wastewater or waste treatment. This is the biggest currently nowadays. So many research institute is focusing on this topic, the biotechnological institute. Techniques involving biosorption process have been found to be promising due to the low cost of non-living biomaterials, which have a potential to absorb metal ions from the wastewater. Now see, this has a very good impact because the seaweeds, the biomass of seaweeds, we can use them against these metal ions or metal pollution. One of the most promising type of biomasses to be used as a biosorbents is the seaweeds biomass, particularly from brown algae. The biosorption capability of seaweeds biomass, okay, seaweed biomass relies on their cell wall chemical composition, okay, cell wall chemical composition, mainly composed of these alginates and fucoidins. These are the molecule with the high presence of functional group that interact with the metal ions and they consume it. This book I have taken it from some book. So these focuses on the use of seaweed biomass for metal biosorption and chemical basis underlying the processes. The current state of commercial status of biosorption technology based on seaweed biomass is presented. Example of complementary uses of algae biomass other than industrial wastewater cleaning processes are presented. And the potential reuse of biomass after the biosorption focused on the biofuel production is generally discussed. So one of the biomass is like Phaeaceae, okay? These large and diverse class of species ranging from small filamentous algae to highly complex organism up to 70-meter long. These class present 2046 species organized into 20 different orders. Phaeaceae is emerged between 150 to 200 million years ago, and it is the secondary endosymbiosis event. That means they possess a secondary endosymbiosis with the metal ions or metal entity, in which red algae was captured by ancestral protist. The characteristic color is given by large quantities of fluoxanthin present in their chloroplast. Okay, this red color is because of fluoxanthin pigmentation. The most important algae order from the point of view of biosorption are fucales and liminarials. Okay, these are the species with a greater structural complexity. The component in their cell wall and reserves polymers have been reported as the main cause of its great capacity for immobilization of metal cation. The cell wall structure varies according to the species, the age of this population, the climate, the geographical location. So everything is actually we need to take care of. The fibril wall structure provides the structural support. It composed of cellulose like a plant. Okay, although displayed lower portion occupying 1 to 8% of its dry weight. Two groups of anionic polysaccharides. Okay, two groups of these anionic polysaccharides that provide strength, flexibility to the cell wall are alginates. Okay, between 10% to, just let me clear these things. Eraser. Yeah. So these alginate between 10 to 40% dry weight, and fucoidin between 5 to 20% in the, which is present in the dry weight. Other components is, these are mannitol, laminarans. They represent the energy backup products that we can use them as a carbon source for our energy generation. So these biomass is also helpful in energy generation. Okay, so other compounds such as proteins or fluorotannins also contribute, but in lesser way to biosorption. They represent actually they represent about 5% of biomass dry weight. This is the algae, that brown algae that we can see. The hyacinths are there, or these Durvelia antarctica. This is the algae that they have extracted from this Antarctica zone. So these particularly, and this is the biomass of that algae, and you can see that these biomass we can use it for different purposes. Okay. Now, next topic in our marine biotechnology is the green fluorescence protein or red fluorescence proteins, which we have isolated from marine environment. So, the first of all applications of these are fluorescence microscopy or live cell imaging. The discovery and development over the past decade of fluorescent proteins from wide variety of marine organisms has initiated a revolution in the study of cell behavior by providing convenient markers for the gene expression and protein targeting in living cells. The most widely used of that is the fluorescent proteins, that is green fluorescent protein. We have first isolated these protein from the jellyfish. Okay, the name of the jellyfish is Aquaria victoria, and can be attached to virtually any protein of interest. Okay, and still told into a fluorescent molecule. The resulting green fluorescent fusion product can be used to localize previously uncharacterized protein, or to visualize and track down the protein to further understand the cellular event. Suppose if we need to identify once some proteins functions, we can attach this GFP with them, we can attach it, and we can insert that protein in the metabolic pathway. And throughout the cell imaging system, we can actually track down where the protein goes, where it binds, whether it binds on cellular surface level or it involves in internal cellular machinery. So everything we can understand the cellular events. The use of fluorescent proteins as mimically invasive tools or minimally invasive tools for studying protein dynamics and their function has been simulated by engineering of genetic variants with improved brightness, photostability, and expression properties. You can see in the figure how exactly these different suppose this is the cell and the nucleus is been stained with green fluorescent protein. We are not able to see these cells, but we able to see only the nucleus, how they are separating. So with respect to this GFP, we can understand how cell division is taking place. Then we can see one mitochondria only. This is the sperm sample, and within that, how mitochondria are actually separated within it. These are some vimentin or these Golgi apparatus. So different proteins labeled with red or green fluorescent protein, we can understand their activity within the cells. I hope so you understand how exactly green fluorescent protein and red fluorescent protein and how they are useful. The next thing that we need to see is the chitosan. So chitosan is the hard biopolymer with a wide and varied range of application. These hard biopolymer generally mainly produced by crustacean group of marine organisms or invertebrates. Although chitin is one of the most available biopolymers on earth, its uses and applications are limited due to its low solubility. The deacetylation of a chitin leads to preparation of chitosan. Okay, so we can get, once we can get the chitin, we can deacetylate them to produce chitosan molecule. These biopolymer composed of randomly distributed beta 1,4 linked D units and has better physicochemical properties. We can make so many shields for the soldiers or shields for laboratory practitioners or researchers or the person who is working in a very heavy environment. We can save their the whole body out of the danger, and by making chitin based or chitosan based some clothes or such a components. Due to the fact that it is possible to dissolve this biopolymer under acidic condition, it can adopt several conformations or the structure, and it can be functionalized with a wide range of functional group or modulate its superficial composition to specific applications. So chitosan is considered a highly biocompatible polymer due to biodegradability, bioadhesivity, and bioactivity. In such a way, this polymer displays the wide range of application. Chitosan is the promising biopolymer for numerous applications like biomedical fields, like we can use it as a skin, bone, tissue engineering, artificial kidneys, nerves, okay, then liver, wound healing property. With the help of, like with the help of chitosan, we have prepared, at least up till now, we have prepared so many different living tissues. We can use them in the surgery. This biopolymer is employed to trap both organic compound and dyes or for selective separation of binary mixtures. In addition, chitosan can also be used as a catalyst or can be used as a starting molecules to obtain high added value products. Considering these premises, this review is focused on structure and modifications of chitosan as well as the application. The next thing that we need to study is the marine microalgae. Now, what are these marine microalgae? First of all, first question, that do you know that 50% of what we breathe is from the oceans? So we need to thank oceans for our every breath. Why I'm saying this? Because the half of the oxygen in every hour breathe is actually coming from oceans or these microalgae. The potential of the carbon sequestration is comes from the fact that marine microalgae are the photosynthetic, meaning that they require light, water, and carbon dioxide to create nutrients necessary for the growth. So culturing these large swaths of these algae would create a very big carbon sink. Why? Because they absorb heavy amount of carbon dioxide from the atmosphere. So once we decrease the amount of carbon in the atmosphere, might be it is the best route to decrease the global warming in the future. But see guys, CO2 is very important. We don't want to decrease the the necessary amount of CO2 or necessary amount of carbon, but the excessive amount of carbon we need to get rid of it. So this could be the very big future opportunity in front of biotechnologists. This is how large array of these algae has been prepared, or these type of lagoons has been prepared Okay, by this is an just artist depiction. Okay, this is not true, the picture is not true, of algal farms in an arid environment just nearby sea. We can place these and we can get energy out of it. Aside from the carbon sequestration benefits, microalgae could also become a major new source for energy in the form of biofuel. The potential use of algae for biodiesel has been studied extensively nowadays, since it is a renewable or eco-friendly alternative to biofossil or fossil fuel. One of the largest research facilities located in Koana, Hawaii, US, is owned by the company Celana. This center has demonstrated that algae can use photosynthesis to efficiently convert carbon dioxide into important oils and biomasses, which can then be converted into feedstock to produce omega-3 fatty acids like oils or like such minerals, animal feeds, and other biofuels. This is one of the structure how exactly these in future how these algal bioenergy we can produce. These are the photobioreactor where they are getting ample amount of solar energy. Once it grabs the solar energy, the carbon sequestration is also there. The biomass is also producing. Now how we are getting energy out of it? We are getting a heavy or heated temperature out of the algae, and we can produce directly with the help of this electricity generation. We can produce energy. The other thing we can produce is the fuel out of the biomass. That fuels we can use directly for the solar energy generation. Then after processing of biomass, we can produce heavy amount of different carbs or biofuels out of it, or ethanol production can be done, animal feeds, or algae oil or mineral oils that we can produce out of it. Okay. Otherwise, these biomass can be used to produce aquaculture feed or fish feed. So there are different variety of different applications are there of these microalgae. I hope so you understood what I mean or what is the basic marine biotechnology. Thank you very much.